Welcome everyone to week number 3. We're already deep into the term this time, so let's see what we've got going on. Okay, last week we covered pH, buffers, and their importance to organisms. pH is a measure of how acidic or basic a water solution or aqueous solution is. This is very important because there are acids and bases out there in the environment, in life, and organisms like to stay at a neutral pH, and that's why there are so many buffering systems. So buffers are sets of chemicals that act to absorb hydrogen or hydroxide ions that keep the pH at a certain level. These are very important because, like I said, organisms like to be at a neutral pH. This week we're going to learn about diffusion and osmosis and how they work. We're going to learn about different types of cells and viruses and structures in cells and viruses. So let's talk about diffusion and osmosis for a minute. Diffusion is movement of any kind of molecule from an area of high concentration to an area of low concentration until equilibrium is reached. Okay, that makes sense. You spray a squirt of perfume in the room. At first, it is very concentrated right where it was squirted, and eventually it moves throughout the whole room and everybody can smell it. So that's diffusion. Well, one molecule that can diffuse is water, and water moves very readily across membranes. That leads us to osmosis. Osmosis is the movement of water across a semi-permeable membrane, and all membranes are semi-permeable. What does that mean? Permeable means that stuff can move across it, in and out of it, and semi-permeable means that only certain molecules can move across it. Well, water can. Okay, and then we come across some terms: isotonic, hypertonic, hypotonic. What in the world do they mean? These terms all refer to a comparison between two solutions. Okay, so cells live in isotonic solutions usually. And what does that mean? Isotonic means that you have the same concentration of water outside the cell as you have inside the cell. Hypertonic, which is not what this slide is, hypertonic is where you have fewer water molecules outside, but lots of solute molecules like sugars or salt. Hyper, lots of sugar. You get it? Okay, but less water outside the cell. So if you got more water inside the cell, the water is going to come out and into the solution. What will happen to the cell then? Well, the cell is probably going to shrivel up. And if too much water comes out, the cell will die. On the other hand, you have hypotonic solutions compared to the cell. Hypotonic means that you have lots of water outside the cell and less water inside the cell. So the water is going to move from outside the cell into the cell, and something may happen. This is where I get to tell you a little story. Back when I was doing my research, I worked with white blood cells that I collected from different people, and I had a senior microbiology student named Heather who was working with me, and she was counting the cells before we could start the experiment. She was on the microscope right behind me. I was sitting at my desk and she was at the microscope, and she gets all frantic and she says, Janie, I counted one square of cells and there were 4, and I looked at the other square and there were none, and I went back to the first one and there were none there. What in the world? So I thought for a second. I said, Heather, did you put them in distilled water? She said, yeah. I said, Heather, do you remember osmosis? She said, oh. Yeah, she had put them in water, and so the water from the distilled water went into the white blood cells, and they got bigger and bigger and bigger and bigger. They all burst. So that's what was happening with the white blood cells. Heather learned an important lesson, and she was a super bright student, but you know, it happens to the best of us. All right, you're also going to learn about cells and viruses and their structures, and you're going to draw some cells and viruses from a key that you have. And since one virus in particular has been in the news so much, I thought I would talk about it for a minute. So we've all heard about COVID-19 and we've heard about the coronavirus. So what in the world? What's what? COVID-19 is the name of the disease. Co stands for corona. Vi stands for virus, and the D stands for disease. So this is the constellation of symptoms that you get to make this particular disease. You get a fever, you get a cough, you get chest pains, you get extreme fatigue, and there are more symptoms than that depending on the person. That's COVID-19, the disease. So the name of the virus is SARS-CoV-2. It is one of the coronaviruses. It is not the only coronavirus. They're called coronaviruses because when you look at them with the electron microscope, they look like they have a crown around them, and that comes from these spikes that you can see in this picture. Spikes stick off the surface of the virus, and they must be important for something, which they are. I'll tell you that in a second. But let's look back at the name for a second. SARS-CoV-2, that's the name of this particular virus that's causing the pandemic in the world. SARS stands for severe, acute, respiratory syndrome, severe, awful, acute, fast, respiratory, you know what that means, breathing, syndrome, a collection of symptoms. CoV, corona, V, virus, 2, the second one that's been identified. So that's why the name of this virus is SARS-CoV-2. It is an enveloped virus, and that's actually important because it's surrounded by a membrane that it picks up on the way out of your cells, so it's part of your membrane. But because it's surrounded by a membrane that is crucial to its contagion, you can easily break down that envelope with alcohol. And alcohol is found in hand sanitizers. So when people tell you that hand sanitizers are not effective, yes, they are, because alcohol dissolves that lipid envelope of SARS-CoV-2. They're actually fairly easy to inactivate, but there are so many floating around in the air now that we're all breathing in that people are getting very, very sick. We're not catching them before they get into our respiratory tract. It's an RNA virus that is non-segmented. That means it has one piece of RNA. It's a positive strand, which means that it can make its own proteins as soon as it gets in your cell. And these spikes are important for a couple of reasons. Those big red spikes that you see on there that are made by the S protein are important for attachment to your cells. So, and that's what gives the virus its corona look. The other spikes are important for helping the new viruses, get out of your cells. I will put a link to the CDC website in case you want to look up more information, because CDC has accurate scientific information about the virus and about the disease. But I am also here at your disposal if you would like to talk about it, if you have some ideas about it. Email me, post something in the discussion board. Join me in the chat on Sunday evenings. I'll be glad to talk with you about it. This is microbiology. That's what I love. So please, please do. Okay, let's continue on with what you need to do. So the lab for this week, and then I've got in parentheses and also next week. I want to talk about the labs this week. You have two. One has to do with potatoes and iodine, and the other one is a dialysis tubing experiment. So what do you need this week? You're going to have a potato from home, some paper, a calculator, and a timer. Wear your safety gear because you're going to be working with iodine, and iodine is actually kind of corrosive. You dilute it down to put it on skin, and it's still not great for your skin. So wear your safety gear for this. There is at least an hour downtime in each one of these experiments because you have to let the potato sit in the iodine solution for, I think it's 80 minutes. That's downtime where you can do something else, Go eat your supper, go watch an hour of TV, something like that. But anyway, you have downtime. For the dialysis tubing, you have to have time for osmosis to happen. So you got more downtime. Just realize that. As I think back over the lab, I'm not sure that downtime is figured in the time recommendations that they say. So just realize that. Okay, in the last column on page 24, all it says at the top of that column is isotonic, hypertonic, hypotonic. And what that really means, if you put these extra words here, you'll understand what you're supposed to answer. In the tube compared to the solution outside the tube, dialysis tubing, so if you put these words there, it will make more sense to you. The dialysis tubing is a little bit tricky to work with. It is a plastic tubing. It sure doesn't look like it when you get it, and you have to put it down in water, and then you can take your fingers like this and rub it, and that will make it open up. Now my fingers are little and tiny, really tiny. I have trouble making the knots in the tubing. So if you find that you're having trouble with that, just use the little clips that they have in the lab. That's perfectly fine. Let me think what else for the potatoes. Can't think of anything else. If you have questions, let me know. You're also making cells and viruses using a dichotomous key that you have in the lab. A dichotomous key is one where you're identifying something, and the key will always give you two choices. So for example, with viruses, you're going to have two items at the top. Oh, can't get it straight. There you go. DNA and RNA. So which one are you going to pick? Because you're going to make your own virus. Which one are you going to pick? DNA or RNA? Choose one. Okay, go down that way. All right, then that branches. You have two choices there. Is it a double-stranded RNA molecule or single-stranded? So you see how it goes. You have two choices, pick one. And I'll talk about the final project in just a minute. So those are the experiments that you have to do for this week, and you're going to make some drawings with those dichotomous keys using the information in the keys. One heads up about week 4. Week 4 experiment has to be started by Thursday in order to germinate the peas that you need to do the experiment with. Okay? And you have to germinate the peas with distilled water or filtered water or water in a water bottle. You cannot use water out of a tap. If you do, and that is chlorinated water, you will kill the peas, and that's the end of your experiment. So make sure you use non-chlorinated water and set yourself aside time before Thursday to get those peas started. Now, there's not much that you have to do to get them started, but you have to start them so that they're germinated in time for you to actually do the experiment. Okay, wanted to mention that. And then finally, you're also going to start your final project. Milestone 1 is due in week number 3. You're going to choose your research question. Remember, I've already mentioned this. Go to Ask a Biologist website. It's in that rubric, in that information. There are 4 suggested labs that you can do. I've also mentioned a couple of others. The breaking proteins one is good. That's a really good one. And the virtual pocket seeds, that's a good one too. There's some in there that are not appropriate because they take too long. You have to order a bunch of stuff. One of them is you get ants from your house and you bring them inside and watch them and do experiments and stuff. I don't suggest doing that one, especially down south where we have fire ants. Yeah, I don't suggest doing that one. So those are my suggestions for labs that you can do. You can design your own experiment. If you do that, you have to get approval by me. So let me know early on because you have to do some research and turn in this milestone 1 this coming week. So the background information that you are going to be looking up and thinking about first is this. So I phrase these in the form of 2 questions. Why is this idea important in the grand scheme of things? So for example, the first lab under Ask a Biologist is the nerve lab, and you're trying to see how many pain sensors there are in different parts of the body. So compare how many pain sensors there are on your back compared to the tip of your finger, for example. Well, why in the grand scheme of things would it be important to know how many pain sensors there are on different parts of the body? Can you think? A lot of young people now have tattoos. Now, if it were me and I was going to get a tattoo, I would pick an area of my body that was not going to hurt like heck to get a tattoo. So probably my back. Yeah, that's probably where I would get a tattoo if I were getting one. So see, I mean, something as simple as that is one reason that this would be important, and there are lots of other reasons. Used to be when you gave blood, back a long time ago, they had to prick you to check your hemoglobin to see if you could donate blood, if your hemoglobin was high enough. And I always loved it because they pricked the side of my earlobe right there. Just pow, little pinch right there, little jab, and they get the blood. And then they all start doing my fingertips. Okay, your fingertips hurt because you have lots of pain sensors there. Your little earlobe right there, not so many. That was way better. So anyway, why is this important in the grand scheme of things? Also, what is known about this phenomenon? I told you in the scientific method that you have to do a lot of background research to figure out what everybody knows about this subject before you get started with an idea and then experimentation. So what is known about pain sensors? What is known about ice formation? What is known about caterpillars and how fast they develop? So what is known about this phenomenon? You're going to be doing this research, and that's what you're going to be doing in milestone 1. All right, that's a lot, but you have a lot going on in week 3. I hope you have a wonderful week, and as always, if you need me, if you have problems that are coming up, if you have a question about the lab when you're doing it, I don't know that I can get to you, right back to you, but I'll get back to you as fast as I can. Email me, chat with me on Sunday night, let me know that you're having trouble, and I will try my best to help you. That's my job here. And not only is it my job, but that's what I love to do. So ask me for help if you need help. I will put my email address once again in the announcements for you. So have a wonderful week.